will ask you a few questions. What is a lymphocyte clone?
A group of the cell
A group of the cell.
of the same epitope or the
Same
same
epitope?
BCR.
No.
Same BCR.
Same BCR. What do you mean by same BCR? Thank you.
That have the same receptor against same epitope.
Same BCR against same epitope. Thank you. So a group of cells. A group of cells. That's all of them have same BCR. It means they have same specificity. All of them are against. How many clones of lymphocyte do we have?
More than 10.
10 to millions of. How this huge diversity is made? Two. One of them, please. Combinatorial diversity. Another one. Junctional diversity. By addition or deletion of some sequence of the DNA in the binding site of the fragments. Yes, thank you. And can a single B cell produce BCRs with different specificity?
No.
A single B cell always have same specificity. When we talk about the population of B cells, it means that we are talking about millions of clones. But a single B cell have many BCRs, all of them have same specificity. Yeah? Can a single B cell produce different classes of antibody?
Yes, can.
Yes, can. By binding to different or diverse constant regions. And she asked me a very good question. Why do we need same specificity with different constant regions? What was it the usefulness of it? Yes, each class has a specific function. For example, when we need a parasite, we need to make IgE. When we need to induce immune response in mucosal surfaces, we need to make IgA. And in primary immune response, we instantly will produce IgM and IgD. What is the difference between BCR of a naive B cell and effector B cell? What is the difference between the receptors or BCR of a naive B cell with the BCR of an effector B cell or memory B cell?
Effectors, B cells have experience with different antigens, different.
Yes,
epitopes.
this is the definition of effector and naive. Effector is antigen experienced, but naive is not antigen. But what is the difference between their BCR? The question is this. You know, you should focus on the question. The question in this, what is the difference? We have an effector and a naive B cell. What is the difference between their receptors?
The BCR, the non-cell have just IgM and IgD,
Mm-hmm.
and the vector of the
Different.
all classes.
No.
Out.
So the naive B cell, thank you. That was correct answer, but with a little correction. So naive B cells always have IgM and IgD, both of them simultaneously. But effector T cells or memory B cells might have one other classes, not all of them, based on the situation, based on the type of antigen. It might have IgE, IgA, or IgG. Okay, so effectors have IgG, IgM, or IgA, one of them, just one of them. But naives always IgM and IgM. So now, after talking about the B cell development, now I'm going to talk about B cell activation. Naive B cell. That's why it's written IgM positive, IgD positive. So whenever, if I give you a question that we have few B cells, there is a question here. We have few B cells. One of them is IgM, IgD positive, another one is IgA positive, another one is IgG positive, another one is IgE positive. 4 choices you have here. We have 4 different B cells. Okay? Salam alaikum. No problem. Can you see here now? Yes, yes, yes. Do you have problem with the lights? No?
No, it's alright.
You have problem? Haji, they have problem with lights.
5 minutes.
It's too long, 5 minutes. Just 30 seconds. 30 seconds.
ما لحق اقول لك شلون.
30 seconds is Arabic.
تمام.
يا خدا. 30 دقائق. ثانية.
تمام. ماكو مشكلة. ما راح نشرب.
No مشكلة. سريع please.
شيء في الكم ولا
No مشكلة.
وسلكتت ما طلعت.
Okay, the question is this. Haji, where are you going? The question was this. We have 4 different B cells that they have different classes of antibody on their surface. The first one has IgM and IgD, the second one IgA, the third one IgG, the fourth one IgE. A, B, C, D. Which one is a naive B cell?
A.
The first one, A. Which one is an effector B cell?
B, C, D.
Which one? B, C,
D.
D, all of them. When we do not have IgM and IgD, it means that the B cell has experienced, has recognized the antigen. Thank you, Haji. Okay? Clear? Now I'm going to talk about B cell activation. So we have a naive B cell and it is recognizing its specific antigen. When we talk about a B cell, indeed we are talking about a clone of B cell, not a single B cell. Yeah? We are talking about a clone of B cell. We have several B cells with same specificities. And as an example, one of them is shown here. Okay? So, a B cell is recognizing its own specific antigen. After recognition, what is going to happen?
Switch classes.
Proliferation. This process is called clonal expansion. Because the members of the clone is increasing as a result of proliferation. For example, the numbers are just example. If we have, for example, just 50 B cells specific to this antigen, after proliferation or clonal expansion, we will have thousands of B cells against that antigen. Because you know, microorganisms proliferate rapidly. Tumor cells proliferate rapidly. So we need immune cells to proliferate, to expand their number rapidly in order to be able to fight with tumor or microorganisms. Okay? The function of immune system is to fight with, for example, bacteria or virus or fungus that you know all of them proliferates rapidly. So we need a huge number of the cells of one clone to be able to fight with this antigen. So the first step is clonal expansion. And there is another term here. How many clones do we have in the body? More than 10 millions. More than 10 millions clones we have in the body. And now one clone is activated. This is called clone selection. Selection of one clone from millions of clones. That's because just one clone is specific to this epitope. After entrance of antigen, we have clone selection. Clone selection means selecting of one clone to become activated from millions of clones. Because just that one is specific to that epitope. So after recognition, clonal selection, we will have clonal expansion. Where is happening now? Where is it happening? No. Secondary lymphoid organs. Initiation of immune response is inside secondary lymphoid organ. We are, for example, it's in the spleen or lymph node. After clonal expansion, we will have differentiation to plasma cell. Plasma cell is indeed effector cell of B cells. And then plasma cells will produce a high amount of IgM. That only IgM, because this is primary immune response. This is the first time that when we say naive this B cell, it means that it hasn't recognized its specific epitope until now. So it's the first time of this cell that is recognizing the antigen. So after the recognition, we will have primary immune response. In the primary immune response, the only antibody is produced is IgM. And over time, after 1 week or 10 days, if the infection lasts, or in the secondary immune response. But maybe in over time, it's better to say over time. After 1 week or 10 days, or sometimes 2 weeks, we will have T cell help for B cell. Helper T cells, do you remember?
Yes.
A group of T cells as T helpers will provide help, will help the B cells. So when we have help of T cell, the nature of the response will change. Okay? When we have the help of T cell, the nature of the response will change. Does specificity change?
No.
No. Specificity won't change. Because the antigen is this. If the specificity changes, so we won't have any response against this antigen. What do we mean by changing of the nature of response after T cell help? The first one is class switching. Changing of the class of antibody from IgM to IgG, IgA, or IgE based on the type of antigen or pathogen. Do you remember I told you based on the type of PAMs and DAMs, innate immune cells will decode the antigen and they will produce some cytokines, and that cytokines will produce the type of T helper cells, and then we will have different cytokines. And based on the type of cytokines, we will have different classes of antibody. So the classes of antibody depends on the type of antigen. If the antigen, for example, is an allergen, the antibody will be IgE. If the antigen is, for example, an antigen in the mucosal surfaces, IgA. Or a protein in the circulation or body fluid, IgG. And they have different functions. That's what I'll talk about them. So, with the help of T cell, the first phenomenon is class switching. In order to optimize the part, this happens because the immune system tries to optimize the response. For example, if for the rest of the response, the only IgM is produced, IgM cannot pass through the, for example, placenta. Or IgM cannot have an effective response against worms, for example. So it is indeed optimization of the immune response over time with the help of T cell. Another phenomenon is affinity maturation. What does affinity mean? Ability of what?
Attachment or bonding or
ability of binding. Yes, we can say. What else?
Ability of binding the antigen.
Ability, the strength of binding.
Yes.
The strength of binding. You know the strength of binding of antibody to epitope, strength of binding. This is important. For example, before affinity maturation, the antibody binds to this epitope loosely with lower affinity. But after affinity maturation, it will bind to them very strongly. It means that it has higher affinity. So the strength of binding of antibody to the antigen is said affinity. So affinity maturation means that we will have antibody that they bind to the antigen more firmly, strongly, and a strong binding to the antigen will increase immune response, will improve immune response. So, question, the affinity of IgM is higher or IgA? Affinity, I'm talking about affinity. Let me ask the question in another way. The affinity of antibody in primary immune response is higher or secondary immune response? With the help of T cell. When there is the help of T cell. I'll talk about it again in the primary and secondary immune response. Another phenomenon is production of memory B cells. So those three phenomena, all of them needs T cells. Class switching, affinity maturation, and memory B cell production. Does carbohydrate can induce affinity maturation? Does antigens that are carbohydrate or nucleic acid, can they induce affinity maturation? Why?
Because we need the help of T helper.
Yes, only for protein antigens we have T cell help. For carbohydrates, for nucleic acid, for lipids, we do not have T cell help. When there is no T cell help, there is no class switching, there is no affinity maturation, there is no memory B cell production. So for carbohydrate, that's why for carbohydrate antigens we mainly have IgM. But for protein antigens we have other classes of the antibody based on the type of antigen. Yeah? That's why we say that I told you we have two types of antigens, T dependent and T independent. T dependent antigens are protein antigens and T independent are non-protein. Carbohydrate, nucleic acid, and lipid. Here is a protein antigen and IgM. This is a naive B cell after recognition and receiving of help from T cell. We will have class switching and the rest of the phenomena and affinity maturation. But for T independent antigens, polysaccharide antigen, there is no B cell head, T cell head, so only IgM, low affinity, here class switching. Low affinity here, high affinity, and short-lived plasma cells. But here, long-lived plasma cells, memory-like plasma cells. I've talked about this figure, primary and secondary immune response. In primary immune response, we have naive B cells, activation of naive B cells, production of plasma cells. The main antibody is, so in primary immune response, the main antibody is IgM. But over time, we will have production of IgG. Okay? Over time, because over time, if the antigen is protein, we will have activation of T cells and help of T cells, then we will have IgG. And the affinity is lower in primary immune response. And primary immune response is induced by all antigens, including proteins, carbohydrates, and the rest of them, but secondary just for proteins, because we have memory just for proteins. Not only in the secondary immune response, not only the class of antibody is changed, the amount of antibody is higher. It's important. Both the class of antibody, the affinity of antibody, then the amount of antibody is higher in secondary immune response as a result of those memory cells. That's why we say memory. After, when we have memory, we will have a stronger immune response. A stronger immune response means higher quality immune response. Higher quality means having antibody with higher affinity, having specific classes of antibody based on the type of antigen, having higher amount of antibody. So, do you remember I told you for T cell, for naive T cell activation, how many signals do we need?
Two signals.
Two signals. The first signal by
Receptor binding to the antigen.
T cell receptor and MHC peptide. And second signal, co-stimulators B7, CD28. B7 on the co-stimulators for T cell are only expressed by B cell. Yes, B cell can. Co-stimulators are only expressed by professional APCs or APCs. Co-stimulators only and only are expressed on APCs. I'm talking about T cell activation. So for naive T cell activation, we needed two signals. Say first signal TCR and MHC peptide, the second signal by co-stimulators. That both of those signals are provided by APCs for naive. And here, for activation of B cells, we need two signals as well. First signal is provided by BCR by binding to the microbe. There is no MHC here. MHC is only for T cell. And antigens are free. Free bacteria, free protein, free polysa, anything. Toxins. So, by binding of BCR to the bacteria, for example, we will have the first signal. And second signal is provided by this one. What is this? Complement receptors. Did you remember I told you one important function of complement system is increase activation of B cells. So complement system will increase B cell activation, will increase antibody production by providing second signals. You know that on the surface of bacteria we have complement activation. And some proteins of bacteria complement that they are bound to bacteria. Now they can bind to complement receptors, and then second signal is provided for B cell. By providing those two signals, first and second, we will have activation of naive B cells. I'm talking about naive B cell. The story for memory and effector is different. Another way of providing second signal for B cell is TLR. What does it? What is that? Toll-like, it's an PRR, toll-like receptor. So by binding of toll-like receptor directly to the pathogen, again we will have second signal. And the first signal is provided always by BCR. Clear? There is another way for providing of second signal for B cell activation. The first way was binding of complement receptor to the complement proteins on the surface of bacteria. Another way is binding of TLR on this is the cell membrane of B cell. Binding of TLR, toll-like receptor, on the B cell to the PAMs of the bacteria, pathogen-associated molecular pattern. And again, the first signal by BCR. Question? So, for isotype switching, what is needed for isotype switching in B cells? T cell help, antigen recognition, and T cell help. And based on the type of antigen, we will have T cell and the T cell help, we will have class switching. Here you can see IgM positive B cell means a naive B cell. Whenever you see IgM positive, it means IgM and IgG, it means naive B cell. So we have a naive B cell which is recognizing the antigen. Over time, we will have activation of helper T cell, and we will have a crosstalk, a talking between T cell and B cell. And here you can see, you know that B cell is an APC. Okay? B cell is presenting antigen. This is MHC. This one is peptide. This one is co-stimulator. And this one, so until now, we have some signals from B cells to activate T cells because B cell is an APC. But after activation of helper T cell will express a protein which is called CD40 ligand, it binds to CD40 on the B cell and send a signal. It sends a signal to B cell. So CD40 ligand binding to CD40 on the B cell will provide a signal that it is needed for class switching. So what determines the type of class of antibody? So there is a B cell that is going to decide to change the class of antibody. Based on what the B cell has to make decision, for example, make IgE, IgA, or IgG. By the type of the microbe. How B cell can determine the type of the microbe? By production of cytokines by T cells. So helper T cells not only provide CD40 ligand and CD40, they will produce some cytokines. And type of the cytokines depends on the type of antigen. Do you remember we had a slide yesterday that PAM decoding of antigen, different kind of cytokines. So based on the type of antigens, we will have a specific cytokine, and this cytokine will determine the type, the class of antibody. For example, when we have interleukin 4 mainly, we will have IgE. When we have interferon gamma, we will have IgG. When we have TGF beta, we will have IgA. So based on the type of the class, the type of the antigen, T cells will produce a specific cytokines, and the type of those cytokines will determine the class of antibody. Okay? Or not? Again, a question. If a person cannot produce CD40 or CD40 ligand, what will happen to that person? A person have a genetic deficiency in the production of CD40 or CD40 ligand. What will happen to that person?
We don't have class switching.
We won't have class switching. It means in the blood circulation, we mainly will have IgM, not other classes of the antibody. It's a routine test, is done in the laboratory to check different immune deficiency diseases. So when a patient has deficiency in this one or this one, it will produce mainly IgM, and this disease is called hyper IgM syndrome. hyper IgM syndrome, because there is no signal from helper T cell to B cell. The main antibody which is produced by that patient is IgM, hyper IgM syndrome. And different classes of antibody have different role. IgM mainly activate complement. IgG can act as an opsonine. It can activate complement. It can pass through the placenta from the blood of mother to the blood of fetus. Ig is important in helminths or parasitic infection and in the activation of mast cells in allergy. And Ig is very important in mucosal immunosurfaces. So, in the bone marrow, the development of B cell occurs in the bone marrow, and production of BCRs is made by combinatorial and junctional diversity, and then mature B cells will leave the bone marrow and goes to the secondary lymphoid organ, and inside the lymphoid organ they meet the antigen, and then we will have immune response. This is a lymph node, for example, that in lymph node we have different parts, that B cells mainly are located here, and here is the location of T cells. After coming of antigens, they are efferent lymphatics, they are afferent, and this one is efferent, you know this, I've talked about them. And then one artery and one vein that take the blood to the cortex and drain the blood. So it doesn't matter what that means. So B cells come to the lymph node from artery, and they goes to their own specific location, which is called B cell zone, the zone of B cell in the cortex of the lymph node. And then T cells go to the T cell zone in the procortex. B cells are located in the follicles of the cortex. Follicle, I told you, is a dense population of B cells in the secondary lymphoid organs. So we have B cell, T cell, and a population of B cell in the follicle. So we have several follicles in the cortex. Here is the cortex and T cell in the paracortex. We have a specific kind of dendritic cells that B cells are around them. Then we will have coming of antigen from lymphatic vessels. So the antigens bind to the B cells. We will have activation of B cells and proliferation, clonal expansion. And after clonal expansion, the follicle is called secondary follicle. Before activation, it's called primary follicle, and after activation, it is called secondary follicle. So in secondary follicle, we have clonal expansion and germinal cell formation. So in the follicle, we have activation of B cells. The names doesn't matter. So after clonal expansion, we will have plasma cells, and those plasma cells go to the medulla and they will produce IgM. This is the activation, the production and development of B cells occur in the bone marrow, but the activation occurs in the medulla. They will produce IgM, and IgM will leave the medulla and go to the site of infection. And over time, if the antigen is protein, we will have the help of T cell. So both T cells and B cells are activated, and then we will have the help of T cell. After help of T cell for T-dependent antigens, we will have class switching. We will have somatic hypermutation. Somatic hypermutation is made to increase the affinity of antibody. Okay? It's another name for affinity maturation. I'm not going to the details. So we will have higher affinity antibody and some memory B cells if the antigen is T dependent. So I talked about B cell development in the bone marrow, B cell activation in the secondary lymphoid organ, now B cell function, or indeed it's better to say antibody function, because antibody will produce, B cell will produce antibody, and antibody fights directly with the pathogen or any antigen. Let me skip the images. This is a tetanus, which is caused by tetanus toxin, and we have vaccine. Most of this vaccine, when we receive this vaccine, our body will produce antibody against a neutralizing antibody against toxins of tetany, and different for in diphtheria as well. And again in polio, we receive poliovirus vaccine, and again we will produce antibody. So in most of the vaccines, polio, tetanus, hepatitis A, B, most of the vaccines that now are being injected, they induce production of antibody, mainly neutralizing antibody. Sometimes antibody will neutralize the virus, for example this one, neutralization of the virus, and sometimes we have neutralization of toxin, okay, or bacteria. You know this, I'll talk about it again. So one function of antibody is neutralization, which can be done both in mucosal surfaces and serum. In mucosal surfaces, neutralization is done mainly by IgA. In the serum is made mainly by IgG. So one function of antibody is neutralization. What is neutralization?
Bound the antigen, bound the antibody to the antigen and then provide.
To the virus, bacteria, or toxin.
Yes, and prevent them to bind the receptor, the own receptor on the B cell.
Yes, it prevents the infection of new cells or more cells. So binding of antibody to the bacteria or virus prevent the binding of bacteria and virus to their own receptors on the target cell. Another function of antibody is opsonization. What does that mean?
Make the antigen delicious or make the antigen delicious?
Making the antigen delicious or facilitating
Uh,
phagocytosis, making phagocytosis easier for phagocytic cells, or improving the activity of phagocytic cells. You can see here that we have a bacteria and IgG, which is bound to the bacteria. On the phagocytic cells we have, what is this? No, this one is antibody. Fc receptor. They are Fc receptor. So by binding of Fc receptor to the antibody which is bound to the microbe, we will have some signals that improve the phagocytosis and killing capacity of the phagocytic cell. Another function of antibody. So when we talk about function of antibody, indeed we are talking about the function of humoral immune response. When we talk about humoral immune response, indeed we are talking about antibody, because the final outcome of humoral immune response is production of antibody. And everything that humoral immune response, or most of the things that humoral immune response can do, is production of antibody. Another function is complement activation. And as a result of complement activation, we will have, tell me the consequences of complement activation. Lysis, yes. Lysis of the target cell or by microbe. Opsonization, complement proteins are opsonin as well. Activation of inflammatory response, yes.
Inflammation.
Yes, activation of inflammatory response, thank you. What else? Lysis, I told. Lysis, opsonization, activation of inflammation, and two others.
Immune complex
Clearance of immune complexes. This one is And the fifth one, activation of B cell. In the previous slide you saw activation of B cell. B cell activation or stimulation. They are the function of complement. Complement activation sometimes is named CDC, complement dependent cytotoxicity. This is another name for complement activation. And after the activation, we will have holes on the membrane of the bacteria and lysis of the bacteria or other cells. And if the viruses go inside the cells, so we have the virus on the surface of cells, then when each cell that is infected with a virus, it will express the antigens of the virus. After expression of the antigen of the virus, B cell will produce antibody. Antibodies are bound here, not there. And then we will have NK cells. That NK cells have, what is this? What is this? What are those? Fc receptors. NK cell will, after binding of antibody to the antigen, NK cells can bind to that antibodies, and then we will have activation of NK cells and release of the content of the granules of NK cell. What do have NK cell inside its granules? What? Perforin and granzyme. That perforin makes hole and granzyme go inside the cell and induce apoptosis of the target cell. When the target cell is killed, then the virus doesn't have a location to proliferate. So another function was ADCC. And they are eosinophils. Another function of antibody is activation, production of IgE against worms or parasites. And when we have binding of IgE to the parasites and worms, we have eosinophils and mast cells, that those cells have Fc receptor for IgE. Then by binding of IgE, and they have Fc receptors for IgE again, we will have activation of eosinophils and mast cells, and those cells will release the content of their granules on the parasite, and then we will have this in the parasite. So another function of antibody is providing activation of mast cell and eosinophil in parasitic infection or allergy. So activation of, I've talked about those things several times. So activation of eosinophil and mast cell is made by IgE. Activate neutralization is mainly by IgG, but IgM can do as well. Optimization IgG. You can see here that IgG has more capabilities, including neutralization, optimization, complement activation, and ADCC. But IgM just complement activation and a little neutralization. And IgA and IgE just one function for each one. So, which antibody is more capable than the others? So it's the end of the story of. I think it's the end story. Yes, it's the end of the story of B cell, and then I will start the story of T cell. Get back to the class in 10 minutes. Please, 10 minutes.